The first talk is by Gemma Jimenez from Sorbonne University, and she's going to talk about the marine biological stations of the French National Center of Marine Biological Resources. So please, Gemma, go ahead.
Thank you, Adelino. Here I go. Can you see my...
Yeah, perfect. I can see and hear.
Cool. Well, thank you very much, Adelino, for organizing this and giving us again the opportunity to present the French research infrastructure and the three marine biological stations that are at the very base of this research infrastructure. I am Gemma Jimenez. I am the Executive Director of the National Research Infrastructure and the Liaison Officer for the French node in the frame of the EMBRC Europe. And I'm going to present you a little bit, some hints on how are we structured and what kind of services are available in the marine biological stations in the French. Oh, sorry, because I do not, French National Center of Marine Biological Resources. So, as I said, we are the French node of EMBRC ERIC. I presume many of you already know the EMBRC. We are one of the 10 nodes, and France was among the nodes that were from the very beginning of this adventure. At national level, we are also a research infrastructure that has been funded by the Agence Nationale de la Recherche. It's a national research agency that is in charge of managing the fundings and mainly the big funding programs by the French government. So EMBRC France has been funded since 2012. And our structure, in a nutshell, it's we have two operators, two legal entities, which are Sorbonne University, it is the main operator, and the French National Research Council, which is the other operator, but it's mainly, it has mainly delegated the coordination to Sorbonne University. And the services are mainly distributed among the three marine biological stations, which are also cooperated by Sorbonne University and the French National Research Council, which are the Station Biologique de Roscoff in the Atlantic coast, Laboratoire Aragon, no, it's the Observatoire Océanologique de Bagnols-sur-Mer in the Mediterranean coast, and Institut de la Mer de Villefranche, which is also in the Mediterranean coast. Here a map for giving a fast view of the, let's say, the geographical distribution. And also we have a headquarter, a small team based in Paris in Sorbonne University campus. This distribution has a lot of impact on the way how we work, but it also shows the geographical area that is covered by the research infrastructure. Now I go to the core services. All our services, we try to follow a classification, and we have identified two main core services, which are exactly the core, the mission of the research infrastructure. One of them is the ecosystem access. As research infrastructure, our mission is to provide access to the marine ecosystems and provide biological resources. The ecosystem access, part of these services, we have these services in all three stations, and part of it is cooperated. There's a collaboration with another French research infrastructure, which is the French Oceanographic Fleet. So how we provide access to these marine ecosystems? It can be through the use of different oceanographic or coastal boats or ships. It can be, here I put you two examples of the size of the ships. They are not big, massive oceanographic ones, but they are very useful for the kind of activity that we do, which is mainly access to the coastal ecosystems. And also with these boats, we have also a set of instrumentation that allows us to collect data of these marine ecosystems. The other main way to access these ecosystems is through the diving, the diving teams, which are scientifically and technically experts. And they, here I put a couple of examples of the kind of ecosystems that they can access with the features of the kind of ecosystems that are accessible from the French marine stations. The other core service, it's not a surprise, are the biological resources, providing access to biological resources. And within this is, let's say, the main or more genuine core service of the infrastructure, in the sense that there's no collaboration with other research infrastructures, and also is in the name, in the very name of the research infrastructure, the marine biological resources. And how do we, what kind of biological resources we provide? We can provide access to biological resources collected in situ. Here I put some pictures, there are many. The star one is the sea urchin, no surprise, but basically any biological resource present in the geographical area within the area of influence of the marine station could be collected, providing that we have the right authorizations and we follow the right procedures. Then another backbone of this service provision are the microbiological collections, microbiological, mainly microalgae. These services are not only the collection, not only the provision of strains, they also include the preservation of these strains, the cryopreservation, the culturing these microalgae at different volumes, etc. And let's say that this service is distributed in the 3 stations by the presence of 3 different microbiological collections. The most well-known is the Roscoff culture collection, is the biggest one and the oldest one. More than 5,000 strains are within this collection. It's managed and located in Roscoff. But there are also 2 smaller but very interesting culture collections, which are the Mediterranean culture collection of Villefranche, which is mainly specialized in dinoflagellates, diatoms, mixotrophic or heterotrophic microorganisms. It's smaller in size, but the specialization is very interesting. And the Banyuls bacterial culture collection, which is mainly specialized in bacteria, and also provide the preserving the strain or keeping the strain that can be donated or donated to the collection. Then the other main type of biological resources that are within the offer of the services of the infrastructure are the marine model organisms. Some of them are collected from the wild, like the sea urchin. Some of them, part of the cycle, can be kept in our facilities, like the catfish. No, catfish, no. I'm not able to remember the English name for this one. But you see the picture. And some of them...
Dogfish.
Thank you very much. So I missed the pet. Thank you. So yeah, for the dogfish, part of its life cycle can be kept in our facilities for experimental experimental or developing studies on development. And some other, like Clitia in the left, our aquaria services are able they have mastered the whole life cycle. So we have different types also of marine model organisms available and at different life stages. Another feature quite unique to EMBRC France, to the expertise within the infrastructure, this is not so evident by the pictures, but is the creation of mutants. Some staff within the infrastructure have the know-how how to create mutants of microorganisms, and there are also some mutant strains available, mainly mutant strains from diatoms, and these are managed by a collection which is the only service that is not in a French marine station. This service is actually in Paris, but also we talk about a very, very quite special feature of of the research infrastructure. The creation of mutants, now we are working, or we have also this kind of know-how for other marine organisms, marine biological resources, not only microbiological ones. And another quite unique feature of the research infrastructure is the presence of a collection. We call it biobank in the English classification, but actually it's a collection of strains fixed in formaldehyde that come from different oceanographic campaigns. Here, a picture of the campaigns that have been well curated and classified, and therefore they are available. The users can access these kind of strains. Mainly they are interested in morphological and taxonomic studies. And to my knowledge, is one of the few collections of this type open to, let's say, to external users. This one is the Cotter collection of plankton strains in Villefranche-sur-Mer. So the two core services, another added value to these services, which are already quite complete, quite strong by themselves, is the presence of what we call proximity technical platforms in the same marine stations where these marine biological resources or this access to the ecosystem is provided. These technical platforms, the proximity is in two senses, both geographical, they are in the same station, but also in the sense of the knowledge. These technical platforms know they are experts in working with the kind of samples and the kind of studies related to marine biological resources, which seems a quite simple or evident thing, but it's not that evident. The biological material from marine origin has some special features that requires a highly, let's say, highly skilled staff, but mainly a lot of experience on that. And these platforms, they have co-evolved within the stations with the activity of the research groups. So they know how to deal with this kind of material. So we have subdivided them in 4 types. And some of them, we have a strong link with other French research infrastructures, such as, for instance, the Institut Français de Bioinformatique, which is the French node of Elixir, and for instance, the data analysis and bioinformatics analysis. So one of the biggest or more, let's say, diversified technical platforms are the one related to imaging analysis, from microscopes of different degrees of performance, from the basic microscope to electronic microscope, fluorescent, etc. We have also a lot of expertise on how to treat these images and how to really get the most information from this kind of analysis already in situ. Here I put also how it is possible to make the link between the cytometry, data from cytometry, and link it with imaging analysis, so it can be done already in situ. Another very special feature within the French infrastructure is the quantitative imagery. Here I put an example of a ZooScan, which is an equipment developed at IMEF, at the Institut de la Mer de Villefranche, and it allows the image analysis, but also the quantification of this analysis through quite automated and let's say highly performance way. It is strongly linked to the development of softwares and image analysis, which is also done mainly in this French station and in collaboration with other research groups. But it's let's say the leader is the group in the Institut de la Mer de Villefranche. And here an image of what kind of analysis we have with this equipment. This image, usually there's then a software that is able to identify or make a first identification of the different zooplankton groups. They have other kind of equipments also developed at IMEF that are able to do this kind of automatization at smaller size of plankton. And then the other, let's say, technical platforms, they are mainly the, let's say, simply the classical ones, the ones that allow any a wide range of genetical analysis, from sequencing to metabarcoding to cloning, etc. They are also present in the 3 stations, although they are not overlapping. Actually, each marine station has its own specialization. Then the mass spectrometry, or the other type of chemical analysis. And even in one of the stations, we have the expertise of crystallography, again, a quite unique expertise on marine, on samples of marine origin. And finally, last but not least, as in the video it was already highlighted, a very special feature that facilitates the access to these services in situ is also the presence of what we call supporting services, because they support the access of the user, of the external user, which are working spaces like laboratories, desks, and others, and hosting facilities and catering. These kind of supporting services, they actually allow the interaction of the users with the staff in the station in a quite unique way. And let's say it's not a service located in one of the stations or in the 3 stations, but there's also the training, the possibility of offering training. We have divided it into two types of trainings. Oops, I don't know why it keeps on going on. One is the training in the use of certain equipment or techniques in order to provide autonomy to the user. This is related very specific to the equipment and the person in charge of this equipment or the technique. In some cases, they demand this training. In some other cases, they can offer it if the user needs it. But this is a training that is, let's say, one by one. The official training should be through courses organized by the Long Life Training Service of Sorbonne University. And it is also possible to request the organization of specific users on demand. So we have already an offer of users, but they are offered by the Long Life Training Service of Sorbonne University. But the user can also request if it would be possible, and then we can discuss about this. So I take here some slides that I already presented in the previous Assemble Plus conference, because I think they are still relevant for this one, which are the added values of working in the French marine biological stations. One of the very first one is the historic heritage. It seems something quite folkloric or not so scientific, but actually it's very related to the spirit or the way people work in the station and the experience of being in a French marine station. Last year we celebrated the bicentenaire. In Bagnous, they celebrate the 200 years of the birth of Henry de la Cache-Doutier, who was the founder. This year, in 2022, Roscoff is celebrating its 150 years of foundation, of existence. And in some years, Villefranche will also celebrate an important anniversary, I think. So just to say that this is a story that starts very long before the research infrastructure, but the spirit is more or less still present in the research infrastructure itself. So another added value, it's inherent in being presently in the scientific environment. The user is embedded, is within the station, where you have interactions with the different researchers, the students, technical staff in the station. Here I put a feedback from one of the users, so it's something that is also looked after by the users. And the specialization and the network, the possibility to start networking with people who are also already well known in their scientific domains. The scientific-technical quality is something also very valued by the users, and mainly also the staff engagement with the users' needs. The users are really supported from the very beginning by the people that in charge of welcoming them until the end, until far after the end of the presence in the station. And here, yes, I put also a couple of remarks. Then, as marine biological stations, they are important actors in the development and the smart specialization of the regions where they are located. So the Bretagne, Occitanie, and Region Sud. And they also host several small and medium enterprises, and they have encouraged the development of many spin-offs. The services are always in constant improvement. I put an example last year. I think I keep it because I think it's a good example how they never stop developing, mastering life cycles, searching, trying to be at the cutting edge of the research, and therefore serving, let's say, the needs of this kind of users. Then there's some other, let's say, less evident aspects, but are demanded. It's using the French marine biological stations. It's almost granted. I mean, it's a guarantee that the bioethical and biodiversity and other relevant regulations are respected. We are very aware and very sensitive to ABS issues, 3R issues, sustainability, etc. All the transversal subjects are taken into account. And also this willingness to be at the cutting edge. We currently are working in the augmented observatories within the EMBRC Europe. All the subjects, all the discussions around verification, etc. It's also something that is, let's say, it's not explicitly in the service offer, but it's part of the service provision. And then I put a last slide, I call it larger than the research infrastructure, again, is to emphasize that we are talking about the research infrastructure is mainly supported by the 3 marine stations, but they do much more. They are older than the research infrastructure, and they do much more than providing services. The hosting of visitors for facilitating scientific discussion and exchange, this is at the very core of the mission and let's say the spirit of these stations. Also, all the activities around training students from high school to PhD level and beyond. The hosting visitors usually are postdocs, and all the activities in disseminating and sensibilizing the general public to scientific and research and, let's say, marine relevant issues that I think all 14, we are now participants, all of us know very well what kind of issues are now on the table. So I finish here. I think I've been quite fast, but actually I think I've said the main part of the presentation. Well, I thought I was faster. Only 4 minutes to noon.
No, I
So
think
if
you were
have
perfect on
questions-
time.
if you have
You were
questions, feel
perfectly
free.
on time. Any questions to Gemma about the French marine stations? Well, one of the things I think, well, we pointed out, but I think it's is the fact that they are in very different ecosystems, and if someone decides to do comparative studies, for example, they can go to the Atlantic, to the Mediterranean, and more shallow, deeper ecosystems. So it's quite and very good hosting, of course. I've never worked in any of the stations, but I visited all of them.
Thank you, Adelino. You have highlighted another of the features. The access.
There you go. You can put another paragraph in the slide.
Yeah. Actually, it was part of one of the very first slides, but I usually go boom and forget about this. But yeah,
No, but of course
a
you
wide
cannot
range.
say
Mhm.
you cannot say everything in these kind of things, but I, you know, it's just something that's quite um, well, it's quite noticeable because of they are slightly, you know, not slightly, they are quite different in many ways.
Yeah, even
Okay, well,
the
yeah,
Mediterranean stations, the kind of ecosystems that you can access are very different. Even if
very different.
they are Mediterranean, yeah. Anyway, I will
Yeah,
not go longer because I see Panagiotis ready.
yeah, yeah. Okay, thank you very much, Gemma. To the next talk, which is about another institute in the Mediterranean, and actually also different from the other ones. This is the Eastern Mediterranean. And Panagiotis Kasapidis is going to talk about the Institute of Marine Biology, Biotechnology, and Aquaculture of the Hellenic Center for Marine Research. That's the EMBRC Greece. And so that's, again, it's quite a variety of facilities from marine biology to aquaculture. So please, Panagiotis, go ahead.
Hello, everybody. Adelina, thank you very much for inviting in this nice conference. And thank you also for your effort for organizing it.
If you
I'll
click on the
present
right.
you, yes, I'll present you the Institute of Marine Biology, Biotechnology and Aquaculture, which is actually the Greek, it represents the Greek node of EMBRC. And the IMBC is part of the Hellenic Center for Marine Research. So I'm the liaison officer for Assembly Plus, for HCMR, and for EMBRC. So just a few words for the Hellenic Center for Marine Research, the whole research center. This is the biggest marine research center in Greece and has premises in 3 areas, close to Athens in Anavissos, close to Heraklion in Crete, and in Rhodes Island. And the center has 3 institutes, the Institute of Oceanography, the Institute of Marine Biological Resources and Inland Waters, and the Institute of Marine Biology, Biotechnology and Aquaculture. And also has 2 public aquaria, one in Crete, the Crete Aquarium, and one smaller aquarium in the hydrobiological station of Rhodes. So from the whole center, our institute, a part of our institute participates in Assembly Plus and EMBRC with its facility. So the center has also some big research vessels. You can see them here in this picture, and also has several ROVs, remotely operated vehicles. But unfortunately, we were not able to include them in the services, in the facilities we provide in the frame of Assembly Plus, although at the beginning of the proposal, we did include the ship, the vessel Philia. Then, in the meantime, we realized that on one hand, it was difficult, it was quite expensive, but also it was difficult to set a schedule because this ship is used for many other activities. So sometimes it's difficult for an external user to arrange exactly the dates of its cruise. And also this ship went for the last 2 years a renovation. It was extended. So for the moment, we don't have
Panagiotis, we are still,
yes.
Sorry, we are still on the
Yeah.
first slide. I think you may have to click on the left. Oh, yeah, now you see it. Now we see another one. So you have to click on the left every time you move.
What I'll make it a PDF, so maybe this in this way you can.
Yeah, that could be another option.
Screen, but yeah.
It could be another option if you do a PDF.
Mm-hmm.
Unless you send it to me and I project it.
So let me try. So do you see now the PDF, the full screen? And the different slides? No.
No.
So
if you want, you can send it to me and
I'll send it to
I'll...
you, Adelino, yes. Sorry for
and then
this
I can
inconvenience.
do it myself. See?
So I hope it's not very big, because now this will be another problem. Okay, oh, it's very big, Delino. Well, I'm not, unfortunately, I cannot send it. Anyway, I'll try to present it from just my screen without full. So do you see my screen now? Do you see the slides? Not. Can you
It's
see it?
still in the PowerPoint as it was. Okay, now you can move it.
Okay.
And
Try
put it
also
in
now the
full
full screen. Yes.
full screen. That's it.
So do you see it now?
That's
Full
fine.
screen?
That's fine.
Okay,
Yeah.
excellent. Sorry for all this mess. So, as I was telling you, yes, a part of the Institute of Marine Biology, Biotechnology and Aquaculture, or IMBBC, with its premises in Crete, participates in Assemble Plus and EMBRC. So here you see the facilities of our institute in Crete. So most of the facilities are based in this main building, where there are the aquaculture facilities in the aqua labs. And here next you can see the Crete Aquarium. So our institute is placed in an area which is quite easy to access. It's very close to the capital of Crete, Heraklion, in the north coast of Crete. So it's just 15 kilometers from the airport, and in the area around, it's a touristic area, so there are many chances for accommodation. Although we don't offer accommodation, there are many chances for users to find good accommodation in the area. So just some overview of the institute. We currently have 37 researchers, but 26, 27 of them are actually elected researchers. The rest are postdocs that also act as principal investigators in projects. So what makes unique IMBC within the frame of EMBRC and Assemble Plus? Because this is actually what all of us are looking for our marine stations, what makes us unique compared to the rest, and what we can offer to the users that they cannot find elsewhere. So what IMBC has unique, it's its position. As you see in the map, you see here all the access providers of Assemble Plus. In the area of the Eastern Mediterranean, we are the only institute present just in the middle. So this is our advantage, that we offer access to these ecosystems. And as I told you before, actually, we are just one node, EMBRC GR, but we are also one operator at one site. So this is the only chance through EMBRC or Assemble Plus to access the Eastern Mediterranean Sea. So IMBBC offer access to quite distinct and unique ecosystems of the Eastern Mediterranean Sea, which is quite oligotrophic compared to the other areas of the Mediterranean as well as the Atlantic Ocean. And then the users can have access to different habitats like macrophyte meadows, Posidonia mostly meadows, and algal meadows, as well as different types of soft and hard substrate. Also, this area is quite unique because it is highly impacted by the ongoing marine invasions. There are many invasive species coming mainly through the Suez Canal from the Red Sea, the so-called deception migration. So the ecosystem in this area, in the area of Crete, around Crete, but in the whole Eastern Mediterranean, is changing rapidly. Many new invasive species appear every year, and the ecosystem of 10 or 20 years ago, it was quite different than the ecosystem of today, and the ecosystem of today will be quite different from the ecosystem of the next 5, 10 years, given also the climate change, where also Eastern Mediterranean is a hotspot for climate change. So there are unique opportunities to study this ongoing process in this area. So this is just a short overview of the facilities we offer in the frame of the Assemble Plus, and I'll go more into detail. So regarding ecosystem access, as I told you, we offer access for the moment to coastal ecosystems around Crete. So we have an inflatable boat that can be used, and also we have a good scuba diving facility with an excellent team of expert scientific divers who have assisted a lot of users, and also we can provide sampling equipment for sampling on different substrates, on the water column, hard and soft substrates. I have to say here that the ecosystem access is the service most asked for us in the frame of Assemble Plus. And also we provide biological resources, mostly species that are collected upon request from coastal habitats around Crete. Here, there are many users that they also ask for fish samples to be collected, although this is not a standard service we provide. In many cases, we can assist the users to also collect fish, but mostly we can collect invertebrates and algae of the soft and hard substrate. Also, we have a small collection of biobanks of DNA and tissue collections that we want to organize better and expand. And also we have expertise in biological sample identification, mostly in invertebrate taxonomy, so we can combine this expertise with the biological specimens collected for the users. Another interesting thing is that now we just obtained funds to construct a new culture collection and preservation facility, having as a model the facility of Roscoff. But in our case, we want to focus on the rich biodiversity of the Eastern Mediterranean Sea. And actually, we collaborate with Roscoff and we use its expertise. So this is a nice collaboration between two operators of EMBRC and knowledge transfer from one to another. So I hope in the near future, in the next 3 to 5 years, we'll be able to have all the facilities constructed and operational. We also have some experimental facilities, some dry labs. There's a state-of-the-art genetics and molecular biotechnology lab, which is the biggest lab of our facility and assists many disciplines and many projects. And also we have 2 labs of marine ecology and biodiversity. In experimental facilities, we also have aquaria tanks. We have very good facilities for aquaculture research. It's one of the best in Mediterranean, I think. But the problem is that most of these facilities cannot be offered within the frame of Assemble Plus because they are used for service provision to the private sector and for experiments of the researchers of IMBBC. But there are a few tanks that have been offered to users within the frame of Assemble Plus. But also most of these facilities can be offered through another TNA project, AquaExcel. So most of the use is done by external users through AquaExcel projects. We also have some very good technology platforms. One is our DNA sequencing platform, which has all 3 generations of DNA sequencing instruments, from the first generation of the standard Sanger sequencing ABI 3730 to the MySeq Illumina and to Oxford Nanoboron. And of course, we don't have only the machinery, we don't only perform sequencing, but we have all the expertise to prepare the samples for sequencing. So we also offer to users expertise and help for library construction and sequencing of amplicons, transcriptomes, DDR, the libraries and small genomes. And also for MinION nanopore, we offer also expertise for library construction and sequencing. Here in the picture, you see our colleague, Dr. Jon Ben Christoffersen, which is our chief technician, and he's very good in operating and standardizing all the procedures for this technology. We also have a small bioanalysis and biotechnology lab with HPLC analyzers, but the machinery we have there is not so advanced, so we can offer at the moment only targeted analysis of biomolecules and metabolites and some bioassays. I have to say that these platforms, bioanalysis platforms, are for structural and chemical analysis, are the ones that have a high demand within EMBRC and Assemble Plus. But as I told you, for the moment, we offer only targeted analysis, and in the near future, we do want to upgrade our facilities so we will be able to provide also untargeted analysis of biomolecules and metabolites. One unique technology platform we have, I think it is unique within EMBRC and Assembly Plus, is our imaging facility that contains a micro-CT scanner. This is a scanner that can scan small samples up to a couple of millimeters. It's a non-destructive three-dimensional imaging technique similar to the computer tomography used in hospitals, and can have very good applications in taxonomy, where you can scan your sample, both external and internal hard structure, keep the image, see it from different angles and perspectives and different levels, and also store it and compare it to other samples without actually destroying the sample. Also, there is a micro-CT virtual lab where many images of different organisms have been stored, and this micro-CT virtual lab is offered to users, but it has been constructed within the frame of another research infrastructure, LiveWatch Greece. Last but not least, we also provide e-services. We have a quite good high-performance computing cluster named Zorbas, which consists of 400 cores and 5 terabytes of memory. It was recently upgraded, and we have installed more than 200 state-of-the-art software suites for research analysis, which are mainly focusing on bioinformatic analysis of NGS data. So also we have constructed many pipelines. Researchers of our group have constructed pipelines, and they are on the server and can be used, for example, for transcriptome and small genome assembly, but also there are tools for gene expression profiling, DDR data analysis, and DNA metabarcoding of prokaryotes and eukaryotes. So as Gemma said in her previous talk, and I just added while Gemma was talking, these proximity technical platforms, because I had the concept, but I didn't have the name. So yes, this is also very important, that most of the services we provide as IMBBC, as EMBRC Greece, is not actually unique within EMBRC and Assembly Plus. But their combination and their proximity, as Gemma very nicely said, gives some extra added value to the user. So we can combine the access to unique ecosystem of Crete, and then the state-of-the-art facilities we have here, the high expertise we have in many disciplines, bioinformatics, genomic analysis, taxonomy, image analysis, and so on. And also another asset we have here in Crete is the nice environment we have. So nice climate, nice sea and sun. So for a user is a nice place to come and work. So I think, although we are not a very big node of EMBRC, I think we can offer to users great opportunities for advances the research. Also, I would like to stress that as IMBBC, our institute participates in many other research infrastructures and research projects. So one field that we are really working on is all the interactions between the different research infrastructures. So we are part of EMBRC. We have the national research infrastructure, which is actually based on EMBRC. It's not actually identical with EMBRC Greece, because it includes also other partners. It's called CMBR. Under the umbrella of EMBRC, we participate in different TNA projects like Assemble and EMBRC, and also we are part of other research infrastructures like LiveWatch ERIC, ELIX in Greece and Bioimage in GR, and we participate also in other projects that are related to research infrastructures. So just before I finish, to present you a couple of success stories where I would like to stress this proximity of technical platforms. So I'll just show you a couple of users where they try to use many of our platforms, because in many cases we have users that they come, they want to collect samples, they take the samples, they just pre-process them, and then they go back to their institutions to complete the analysis. But in this case of this user, Dr. Yorune Sakalauskaite from the University of Torino, she was studying, she wanted to do multi-omics analysis of a gastropod of an oyster, the Spondylus gaideropus. This is an important gastropod also for archaeology. And the aim for her visit was to obtain the transcriptome from the cell-forming mantle tissue of this species. So we assisted her in collecting live specimens of Spondylus gaideropus, extracting mRNA and genomic DNA, and prepare here the libraries for mRNA for transcriptome sequencing that she later performed in her institute and with other collaborations. We also had a similar case, but in another DNA project, EMBRC, where the user Isabel Cunha actually performed the whole analysis here, from collecting the samples of a barnacle Lepa sanatifera, doing the RNA and RNA extraction and the library preparation, and then also the bioinformatic analysis for transcriptome assembly and gene characterization. She was assisted because she didn't have this know-how, and then the end was also a collaboration that produced a paper that you see here. And this is another case of another user, Kimani Kitson Walters. He's actually based in the Caribbean Netherlands, in the St. Eustatius Island. So his aim was to compare ecosystems in two areas associated with the Alophilus stipulacea macro plant. This is an invasive species in the Caribbean, but not in the Mediterranean. And sorry, also in the Mediterranean, I think, but not in the Red Sea. So he wanted to compare different ecosystems from the native ones to the invaded ones. So he had access to the ecosystem. He performed field experiments, collected samples, and also processed the samples in the lab. And our taxonomic expert helped him identifying the samples. So that's it for my part. Thank you for your attention, and I want to apologize once again for all this mess at the beginning. So
it was good, Panagiotis. Thank you very much. I think you gave a pretty good overview of the institute and what's on offer. You forgot to mention the food as well. It's also an attraction. By the way, EMBRC and AquaExcel are going to partner, perhaps at the end of the year, beginning of next year, in a joint call for TNA. So basically, yeah, there is some overlap, and of course the facilities that you have are very good for aquaculture.
Yes, this will be very
any
good.
questions?
Actually,
Sorry, you can talk. You can talk.
No, I think this is very important because yes, here in the HCMR in the EMBRC we had these two different TNA projects that they were actually overlapping at some point, but they did not collaborate. So this would be good to bring under the same umbrella actually more people that are working on the same facilities actually. Yeah.
So yeah, that will be something that it will be part of this, EMBRC offering a regular TNA independently of having of European projects or not. But in this case, it will be a joint activity. You've got the video.
Yes, if you have some time, it's 5-6 minutes.
Yeah, you can show the video.
So it's a video we produced for our EMBRC. So just so you see the full screen, I hope.
And I like the name of the computing, Zorbas. Okay, thank you, Panagiotis. I don't know, was there sound on it or?
You didn't hear anything. So,
No, we didn't hear.
yeah.
I think it has to do with the
So,
way it is shared, but
yes,
I think it's on the
yes,
YouTube.
yes.
We
Yes,
can
on the
always go
YouTube. So
back.
if you go to IMBBC, and yes, it has very nice narrative and very
Okay.
professional English.
I wasn't quite sure whether it has sound or not. That's why I didn't interrupt
No,
it.
it had
But
a nice music. So
okay, I'll have a look. Okay, well,
okay,
thank you very much. If there's any
I thank
questions,
you.
just to finalize. If not, thank you very much, Panagiotis, and everyone that has been listening. Welcome back. Good afternoon, everyone. So here we are again for the Assemble Plus conference, and it is my big pleasure this afternoon to invite Fritjof Kuipern, Professor of Marine Biodiversity in the University of Aberdeen, to give us a talk. We know each other for 20 years, I think, now, because we started it started in Marine Genomics Europe quite a while back. And yeah, it will be great to listen to your talk on scientific diving as a research tool. We actually, diving is one of the services that are offered throughout marine stations, and it's becoming really very important, more and more important. So please, Fritjof, thank you very much. Go ahead and give us.
Thank you, Adelino. Yeah, I'm absolutely delighted to be here. Assemble is like family for me. And as you say, I mean, we've known each other for a very long time, and we were involved in writing the first Assemble proposal. I remember the meetings in Naples and Oban and other places. And yeah, and as you say, diving is a tool which is available at a number of Assemble sites. At some point when we had Assemble meetings moving from site to site, I actually tried to dive at each of these sites to see how the facilities operate. I did not manage to dive all of them, but most. And but now I'm going to say a bit more about the potential of diving as a scientific tool. So I'm going to share my screen here. You should be able to see my presentation screen. Okay, I'm now in full screen mode. You should be able to see this.
That's fine. That's fine. Perfect.
Yeah,
beautiful.
Underpinned, I should acknowledge, with photos from our various expeditions around the world. And this, in particular, this photo was taken by Hugh Brown at the former UK National Facility for Scientific Diving, which is now Tritonia Scientific in Oban, still part of an Assemble partner. And diving as a technique is rather young. For scientific purposes, it is about since the 1940s, and it was pioneered by Jacques-Yves Cousteau and Hans Haas, and mostly in warm seas, in the Mediterranean and the Red Sea. However, as we well know, marine biology has started much earlier, and arguably in ancient Greece and Rome, and I should mention names like Theophrastos and Pliny and others. And there was a new dynamic in the 19th century in the wake of Darwin, with people like Hooker Dorn, who was also the founder of the Stazione Zoologica in Naples, Haeckel and others. But diving has the unsurpassed advantage of putting the scientists right into the environment or the communities that are studied. And a key point that I'm going to make here is that being able to see, touch, and selectively collect in situ is a priceless advantage. And this applies especially to the benthic, to the seabed environment. And before diving, all the collections of specimens and samples relied on fairly indiscriminate and often blind methods that you couldn't see, because those days you had no live video link to the surface from some ROV or bottom lander. Which means all the sampling was pretty much haphazard. You dropped some device, it grabbed down, and it was then a bit of a lottery what you were about to pull up. And yeah, such devices are dredges, grabs, and coras. And yeah, consequently, this applies now to my field, which is algal diversity and algal biochemistry. The algal diversity below the tide line remains until now actually very poorly known, and it is biased by mechanical sampling methods. And even in the Mediterranean, we can say this, we know actually very little about the algal flora beneath 30 meters depth. And that applies even more to remote places in the world, like the Falkland Islands in South Georgia, where a Swedish pioneer, Carl Scottsberg, worked around the 1900s. But he missed, with all respect for him, and he did monumental work, but he missed part of the deep water flora. And that's because collections at that time were only possible by dredging and bottom grab. And he probably would have dreamed of the possibility of diving in these places. And the same applies for Ascension Island. That's another very remote island in the middle of the South Atlantic that I'm going to talk about today. And yeah, this is where our diving-based expedition with 14 days of diving, almost exactly 10 years ago, 2 psychologists, one myself, yeah, yielded at least 15 new records in only 14 days of diving. And the same applies for what other teams did within the framework of the same expedition who were looking for fishes and invertebrates. And also the point has to be made that the quality of samples that is collected by diving is often unsurpassed by what mechanized tools can achieve. Because you've basically a diver with a precision instrument, which are his hands in the environment that you want to study. And in many cases, yeah, the quality of such samples is so good that they can be directly further processed by established techniques. And that applies especially to microbiology, culture, molecular and cell biology, and also biochemistry. And there are also environmental sustainability advantages to diving. That is, it is relatively non and low intrusive, low impact. And unlike dredging, which is more like sending a bulldozer into a seabed ecosystem, there's very little interference with what you want to study and very fewer conservation issues. And also diving is quite mobile and versatile. It can also often be arranged with short notice or little logistic effort. And in some places in the world, you can use tourist infrastructure to some degree. I mean, I do not want to sort of downgrade the value of scientific diving, but it is a kind of technique that is available throughout much of the globe nowadays. And yeah, another advantage is, of course, the documentation in real time. And this is since the rise of digital photography that you have an immediate result. And that is parallel to collections or an experiment. That is a difference compared to when I started out in the mid-1990s, when you had to develop 35mm film and you waited for a few weeks until you got them back as prints or slides. And yeah, and it goes beyond that. A diver can really become the operator of an experiment here. He can set up, fine tune, follow, and recover an experimental setup. And the capabilities of human hands, eyes, in particular the brain, which are the key tools of a diver, are hard to match by the technology that is available on ROVs and benthic landers. And it is bound to remain like this. In the offshore oil industry, there's a tendency to move away from diving for insurance reasons mainly, but I think these are key aspects which are very difficult to match by technology. And there's so far no ROV onboard computer that is comparable to the cognitive capacity of the human brain. ROVs have their role for specific tasks, but still having a diver in an environment has some unsurpassed advantages. And the main is that the scientific diving puts a scientist's brain operationally on the seabed, within the depth limit where a scientist's brain can operate. In principle, the same argument is also put forward in support of manned spaceflight. And I would argue that overall, with further improvements in diving technology, the scope for scientific diving, actually, I would expect will increase. And it is also relatively low cost. A scientific diving operation is feasible for tens or hundreds of euros, pounds, or dollars. For example, for our Baffin Island expedition in 2009, we calculated that one diving operation cost about £600. That's about at the upper end of what something like this would cost. When working in Ascension Island or Greece or Cyprus, it is probably less than £100 per diving operation, excluding salary. And it is almost impossible to do any serious ship-based work in marine biology or oceanography in that price range. Typically, the cost is about 1 or 2 orders of magnitude higher. Of course, it's another matter if divers require a large research vessel, then it becomes quite expensive. Yeah, and for comparison, again, I mentioned manned spaceflight. It takes a bit over £5 million to put a person into low Earth orbit, which means it's about 5 orders of magnitude higher than a diving operation. So, something else is also that the carbon footprint of doing scientific diving work is much lower than using a research vessel for the same objectives. And again, we're talking orders of magnitude difference. And this in this scenario, it implies taking a diver, and that maybe by plane actually, to some place in the world which is to be studied, and then diving locally using small vessels, as opposed to a large research vessel which will burn tens or hundreds of tons of fuel a day. Yeah, I calculated the carbon footprint of this aforementioned Arctic expedition in 2009, and we're looking at about 500 to 600 liters of aviation fuel per team member, and about 70 liters per team member locally, which means the whole expedition consumed about 5 tons of fuel for a team of 7 scientists and divers, and that's about the daily fuel use of a rather small research vessel. And we were in the field for about 3 weeks. The main limitations of diving, and there are limitations, are human physiology and the degree of acceptable risks. It is potentially a hazardous activity, and here in the UK, it is regulated by the UK Health and Safety Executive, and there's analogous legislation in every country that I'm aware of, certainly within all the assembled member countries. Most of these risks are related to decompression accidents and diseases. In cold seas, like what we have around Scotland and polar regions, there might be hypothermia. There might be dangerous marine life that is often overestimated and overstated. But often these risks are exacerbated by the remoteness of study areas. There's a tendency that the more remote areas tend to be the more interesting ones, especially when it comes to marine biodiversity and ecology. And yeah, again, our 2009 Baffin Island expedition is a good example. The nearest decompression chamber was in Montreal or Ottawa, which would have been at least 2 days of travel away from the study site, and that would have involved several flights, which flying is something you want to avoid when having a decompression accident. So this is a limitation. And therefore, the insurers, together with the health and safety people at SAMS, then imposed a depth limit of 15 meters. But that thankfully actually sort of covered most of what we were studying. So we were quite happy with this. Other limitations are certainly the level of diving technology and reliability and performance of equipment that are available. Now, this has improved a lot over the years. And the equipment that we have nowadays is no comparison to what people like Cousteau and Haas had in the 1940s. And maybe also cost. I mean, remote areas or interesting areas in the world often are developing countries. And you may not find the same kind of quality of equipment that you are used to from the first world where we are fortunate to live. Yeah, other limitations certainly are depth, because depth exacerbates the risk of decompression accidents, duration, the time that you can spend underwater, because you usually have a finite amount of air unless you have a surface supply by a hose. Usually it is not temperature, because dry suits can be used in polar regions and they're very well insulated with undersuits. They even inbuilt heaters and things like this. And that means basically every aquatic environment on the planet can be dived. So temperature is not so much of an issue. I'm now going to talk about a few success stories. And yeah, for example, George Pettit, whom I got to know when I was a grad student at the Roscoff Marine Lab in the 1990s. And he has sadly just passed away actually last September. He was one of the pioneers of marine natural products research. He was among the first to isolate anti-cancer compounds from especially soft marine invertebrates. And he has been active since the 1950s. He was a true nestor of the field. And by the time he passed away, he has produced over 800 publications with over 32,000 citations and an H index of 89 and dozens of patents. And in the large majority of these studies, the collections of marine organisms started with materials collected by diving, like this one. That is probably the most famous compound that has come out of his school, called bryostatin 1. And yeah, it is one of the first major promising anti-cancer drug development leads from a marine organism. And initial activity was found against various cancers, including lung, colon, CNS, melanoma, ovarian, and renal cancer. And it is in advanced phase 2 clinical trials in the US and Europe. And of course, when you find an interesting compound from a marine organism, then you have a supply issue. How do you get enough of this stuff? In this case, it was addressed by aquaculture. The source organism is a fouling organism that is a nuisance on many man-built structures in the sea, in the Pacific. And also molecular biologists have been able to clone the biosynthetic pathway genes into organisms that are much easier to culture in the lab at a biotechnological scale. And also then organic chemists came into play, and this is typical, who were able to synthesize analogs, structural analogs, which have nanomolar IC50 values. And that is ideal. This is the dream of a pharmacologist, to have active compounds with such a low active concentration. I'm now going to show something from our work in the Arctic. This is already quite a while ago, but I'm currently trying to get funding to go back to this interesting part of the world. And you're all familiar that Arctic sea ice is shrinking rapidly. And the place that we visited is Baffin Island. That's one of the largest islands in the world in the north of Canada. It's about 2.5 times the size of Great Britain, but hardly anybody lives there. This is why it's not on the public mind. There are only about 11,000 inhabitants, and most of them are here in this capital, Iqaluit, and we worked in the vicinity of this remote Inuit community called Pond Inlet in the north of the island at a place called Cape Hat. And yeah, this 3-week expedition produced over 10,000 images and over 20 hours of video footage and hundreds of herbarium specimens and numerous records of algal pathogens, about 70 live algal isolates, and also about 200 samples of seaweed tissues in CTAB and RNA later buffers, as well as CCAL gel, which was then exploited for a 454 sequencing project. And we also had isolates of freshwater iodites. And yeah, the follow-up work typically continues for many years. Actually, most of it is published, but not everything. We're still working on bits of the materials that we have collected. You know, all the herbarium specimens have been identified, but then we applied what is called the germinal emergence method. And this is a technique which I consider a powerful combination of diving in combination with lab-based techniques. When you go to such a place, you have very limited time in the field, and you will have often very primitive equipment to study what you're collecting. And to overcome this challenge for detecting algal diversity, we applied an approach where the diver will take sterile tubes, Falcon tubes, on a dive, fill them with bits of sand or sediment, and then incubate them after the expedition in the lab. And here I have to acknowledge my longtime friend and collaborator, Akira Peters in Roscoff, who pioneered this. And this incubation in the lab will typically result in dozens or hundreds of algal isolates that you will never see during such a short expedition. And in many cases, these are the things that are novel because these tend to be quite small, they're cryptic. And indeed, in the case of this Arctic expedition, we have a number of new records and even species new to science. And yeah, this is how it works. Again, based on the collection of sediment or substratum samples, which are then incubated, you get unialgal isolates, and then they're characterized by microscopic and molecular techniques. And then with bioinformatics, you can de-replicate what is actually novel. And you can focus on what is in-depth studies of novel taxa. Most of this work has been published, so you can find this, it's open access. Another interesting case of this expedition and where diving was essential was the work on Platycyphon and Platyarcticus. These were described as 2 endemic brown algae from the American high Arctic, and prior to this work, it was unclear what is their phylogenetic affinity. And this is what they look like. They're actually quite different. On the left, you can see the Platycyphon, what was previously described as Platycyphon, and on the right, Platyarcticus. When doing sequencing work based on our diving collected material, we actually figured out this is the same thing. And what you have on the left is the juvenile form. On the right, it's the adult form, the multi-year form that has spent a whole season under the sea ice. And yeah, as I said, there was a plethora of video materials coming back, and we produced a documentary about this, which you can watch online. There's a link embedded here in my presentation. It's available online. And yeah, a number of images have been deposited in AlgaeBase, which is the world's reference database for algae. And the diving, you might wonder, what is it like to dive in the Arctic? And the equipment is neoprene dry suits and 2 thermal undersuits. And as regulators, we tend to use either Poseidon or full face masks, which are adapted to the cold temperatures and which significantly do not easily freeze up, which can cause a free flow and emptying your tank in no time. And usually would also have a second cylinder with a regulator for backup. And the standard team was 2 divers in the water who are linked by a rope in the so-called L configuration. And you'll see this in a photo in a moment. And then 1 dive supervisor on the shore and a fully equipped diver on standby ready to go in. And then because it's the Arctic, you also need a shore and boat support team with 1 or 2 rifles because of the polar bear hazard. This is actually quite significant, especially in the summer, because that's the hungry season for polar bears. And as I said, the maximum depth limit was 15 meters because the nearest hyperbaric chamber was 48 hours of travel away, so that was not really an option. But we managed to accomplish dive times of up to about 1 hour. And we conducted about 50 diving operations, almost 30 hours on the bottom, and average max operating depth was 10.4 meters. Here you can see this L configuration. That's Martin Sayer on the right and myself on the left, with the long end of the rope going back to the shore. So much about the Arctic. I'm now going to mention a few other places in the world where we have worked, in particular the Falkland Islands, Antarctica, and Ascensions. This is Ascension Island. It's this speck of dust in the middle of nowhere in the South Atlantic, and then the Falkland Islands, and they're actually linked by the Royal Air Force Air Bridge. This is the obvious way to get there, not obvious to everybody, but it's very convenient. And yeah, we did this first major expedition in 2010. There have been several more since then. And from the Falklands, I then went for the first time also yeah, to Rothera, to the main British base in Antarctica. Then on the way back, I stopped in Chile and the Falkland Islands again, and then in Ascension. And this is where we surveyed actually the seaweed biodiversity of these places. And here are a couple of images. Yeah, the Falklands are one of the most spectacular places where I have dived. It's absolutely spectacular. And yeah, we've last been there for diving in 2017. So it's maybe time to go back. It's already 5 years. And one example of what you can accomplish only by diving and not by using bottom grabs is this. This was overlooked by the pioneer whom I mentioned, Scotsberg. Silurgoderma australe is a brown seaweed that is closely attached to coralline algae, and you're very unlikely to collect it by a grab unless you have a lot of luck. And consequently, it was a new record. And now a few images from Antarctica. This is Rothera. That's the main British base in Antarctica. It's a fantastic place to work. It's a pity it's not part of the Assemble network. I had to think this repeatedly. But there is a nice access grant scheme of the UK Natural Environment Research Council to visit this place. And yeah, that's what it looks like. It's a cold place. That's Dave Smith and myself diving at Rothera. And yeah, this is what it looks like in the summer. It's basically like diving in vegetable soup. For most of the year, Antarctica has super clear water, but this is in the middle of the summer phytoplankton bloom when visibility can be down to about 1 meter. And still we managed to do a seaweed survey. And what kind of impaired us were these guys, leopard seals. They're the top predators in this ecosystem. And whenever they showed up, and there are a lot of them, diving was outlawed. There was no diving. So you actually have a lot of time for other things in Antarctica, and you should budget your time for that. If you want to do a lot of diving work, go to Antarctica with a lot of time, because you can safely assume that about half of the days you will not be able to dive because of these leopard seals. And it's also spectacular for photography. So when you cannot dive, you have time for things like this, like photographing penguins under the midnight sun. So the major outcomes, again, were thousands of photos, 2 species of red algae, which are new to science, and also 22 new records of seaweed species, which were new for the Falkland Islands and Adelaide Island, Antarctica. And this is what I said. Once you start serious diving work in such remote places, you're quite likely to come across a lot of previously unrecorded taxa. And we have published this. And for my second trip to Antarctica, which is now 4 years ago, there was another case of what is the most common and most conspicuous brown alga in that place. It's the main canopy-forming brown alga in that part of Antarctica. And then there was a mystery seaweed that showed up, and I thought, what is that? And then sequencing revealed that this is actually what you have at the bottom is the juvenile form of the old form. The old form on the top panel is very well known for decades, but the juvenile form was hitherto unknown. And we've got an analogous situation of what I just mentioned with these seemingly two Arctic seaweeds, which were in fact one. And then a few words about Ascension Island. Yeah, it's a speck of dust in the ocean. It's likewise a pretty spectacular place to visit. One of the reasons is it has the largest colony of green turtles in the world. Thanks to effective conservation, there are about 18,000 of them, or maybe more by now. And well, we went there for the first ever comprehensive survey of Ascension's marine biodiversity. It was led by my friend Paul Brickel, who is director of the South Atlantic Environmental Research Institute in Stanley in the Falkland Islands. The work was funded by the UK government's Darwin Initiative, and we conducted a macroalgal survey. We, that is Kostas Siamis and myself. And we recorded 75 species, and about 20 of them were new records for Ascension. We have published this work in JMBA. And also, yeah, this EMBRC was just completed. Actually, a few weeks ago, it's not ongoing, it was just completed. Diving in Ascension, you realize that most things are tiny compared to what you will find in most other places in the world, and that is due to intense grazing pressure. This little green thing here in the middle, Neomeris, is about 5 millimeters in size. And a bit further to the left, you can see these blue iridescent dictyotas. I mean, these are just millimeter-sized representatives of algal genera, which are much bigger elsewhere in the world. The diving work by our colleagues from the Falklands also involved these settlement plates, which were deployed and then which are monitored, again, by diving every couple of months. And the main culprits why everything is so small in Ascension are these guys. This is an omnivorous fish, the blackfish, Melanichthys. And actually our expedition, which was focused on algal biodiversity, had a chance finding. We realized actually, and this is only what you can do when you're really a diver, that these guys are cannibals. They eat one another. This was not in the literature. So if you dive with an open eyes and open mind, you may discover things that you would not have expected. And yeah, these are some impressions from Ascension. It's a pretty spectacular place. There are lots of underwater caves. It's linked to the volcanic past of the island. There are lots of turtles, which you see also underwater. And the seabed is dominated by coralline red algae, like these mel nodules and these sessile coralline reefs. I mean, they form reefs. There are no corals of any large extent in Ascension. I'm coming to the end of my talk. More recently, we have worked a lot in Kuwait. I have 2 PhD students from them. One has just finished very recently. And the one who's finished recently was looking at the marine natural products, the chemistry of seaweeds from the Gulf. Yeah, this is us doing diving work. And with the support of the Kuwait Coast Guard, we also surveyed some of the offshore islands, which are not as rich in coral diversity. That's because the Gulf is such an extreme marine environment. It gets very hot in summer. The sea gets up to about 37 degrees, and there are not many corals that can withstand this. It's also actually quite poor in visibility, and that's because of both the silt from the rivers of Iraq, but also the natural eutrophication. And the photo on the left is at almost 36 meters depth. It's pitch black. And that's because of the high amount of suspended matter in the water column. This is a bit of a jump to the Mediterranean, to Rhodes Island, where diving work, again, in combination with German emergence cultures, revealed the second ever finding in the world of this mystery alga called Schizocladia. It's a sister group of brown algae. And we got a culture of this. Again, big credit to Akira Peters in Roscoff. And again, this would not have been possible without diving. And we recently had another PhD in my group about the seaweed diversity of Malta, which was basically a project to get a catalog of the seaweeds of Malta underpinned by molecular data. The PhD of Angela Bartolo just completed. And again, this heavily relied on diving. This is myself collecting materials of Malta. That was 5 years ago. Malta is another very interesting place to dive, very clear water, and actually in many cases, a perpendicular coastal profile. There are spectacular underwater cliffs. Finally, now I'm coming really to the end. A major activity in my group is seawater desalination. This is the applied line of research, and this is because there are arid countries in the world, like the Gulf region, but also much of the southern and eastern Mediterranean, that rely on making drinking water from the sea, and that has marine environmental impacts. This is a large desalination plant that I photographed in Kuwait a few years ago. But currently we have an ongoing EU project focusing on the desalination plants of Cyprus in the Eastern Mediterranean. What you can see here is the outfall pipe, and you can see it's quite turbid. Actually, this is due to the higher density of the brine, of the highly concentrated salt solution, which comes out of this diffuser pipe, and we are studying what that does to marine life. And countries like Cyprus rely to a high degree on desalination. There are other countries like Malta, Spain, and Greece also. The technology they use in the Med is actually quite energy efficient. It takes about 5 kilowatt hours per cubic meter to make desalinated water using reverse osmosis. As I said, we are exploring what this does to seagrass meadows. And I will now show you some photos. We've just published this. Again, this is open access. We found that seagrass meadows are negatively impacted. That is my friend Eleni surveying the surroundings of such an outfall pipe. We also have an observation that we want to explore in more depth. We have the impression that these outfall pipes attract more alien species than pristine environments, pristine sites around them. So this is something to follow up. It seems like they're hot spots for the spread of alien species. Finally, and this is now the end, the most recent project, and this that we've done with EMBRC support, and this is why I still want to mention this, is we've been looking at how kelp forests impact the chemical speciation of iodine in seawater. We were first working in California using giant kelp as a model, and we used diver-operated syringes to take precision samples of water from around these kelp thalli, which were then electrochemically analyzed in the lab. And indeed, we found, in a nutshell, that kelp changes the chemistry of iodine in seawater. And this is really the EMBRC funded work. Yeah, this was done in 2019 in Oban, and I'm actually about to go back to Oban tomorrow, funded by EMBRC, to continue this. Yeah, we found that this applies also to European kelp forests. And in this follow-up project, which is led by yeah, my Greek friend and colleague, Maria Protopova, we're going to look at whether this changed chemistry of iodine is also going to result in a changed zooplankton. Yeah, with this, I'm at the end of my talk. Thank you very much for your attention.
Thank you, Krzysztof. Anyone would like to ask a question? Very interesting and beautiful pictures. I have a question. You mentioned that you got certain species or some species of algae in the Falklands and around the Antarctic continent.
Yes.
Was that it? Somewhere in the Antarctic itself?
Yes. Rothera is the place we've mostly surveyed. Yeah.
It was in Rothera. So is it the same species that you're talking about? It's surprising.
There are big differences between the Falklands and Rothera. The flora at Rothera is, I would say, impoverished compared to the Falklands, and it is quite different because even though these are both cold southern hemisphere places, the big difference is that Rothera has a lot of sea ice for part of the year, and the Falklands do not. And
no, maybe I misunderstood. I thought it was the same species, but probably not, because I think
some are the same, but most are different. I would say most of the diversity between Rothera and the Falklands is quite different.
Exactly. Yeah, because you've got the circumpolar current that stops anything from moving from one side to the other.
It's like a barrier, like a boundary in the sea. Exactly. Yes.
Yeah. Okay. Well, thank you very much, Krzysztof. The next talk is by Erika Becker. She's from the University of the Federal University of Santa Catarina in Brazil, and she's going to talk about macroecological patterns in South Atlantic, brief overview of copepod ecology from a trait-based perspective. So please, Erika, welcome, and it's your turn to present.
So let's start. So hi everyone, I'm Erika. I'm from the graduate program in ecology in the Universidade Federal de Santa Catarina in Brazil. So I'm going to present macroecological patterns in the South Atlantic with focus in copepod ecology in a trait-based perspective. Okay, so well, so trait-based studies are mainly focused in terrestrial ecosystem, mainly use plants as a model. But in the past year, we see an increase in the studies, mainly because of this study of Elena Lichtman in 2013, which overviewed the zooplankton traits, targets some zooplankton traits. And well, a key, the concept of trait is describes an organism's ability to feed, survive, and reproduce. So in this sense, a ground-based support in the studies is try to link species traits to ecosystem functioning. So after this previous work, we can classify plankton functional traits in type, ecological function, and also traits that we can measure from images and the ones that we can infer from images. So in traits type, we have life history, behavioral, physiological, and morphological traits. and which is related with ecological functions like resource acquisition, growth, reproduction, and survival. So when we look to traits, body size is considered a master trait because it goes beyond the different ecological functions, and also because it transcends the different size fractions of plankton taxonomic groups. And also another importance of body size is that we can actually use as ecological proxy to see global change. So this is the very first work which looked into biogeography of key traits of copepods. So using body size, feeding mode, relative offspring size, and myelination, which is a similar structure as a nervous system. So when we look to body size, we can see that there is a decrease with temperatures. So body size decrease equatorward, and this is actually confirmed the temperature size rule. And also this decrease is related with a decrease in productivity, is possibly driven by the length of food web chain, and also there is the size selective predation in warm waters. So when we look to the large scale, we can see that the gradients in zooplankton body size is mainly driven by copepods. This is because they dominate community composition and abundance. And we also can see some patterns. For example, there is a difference in the north and south hemisphere. So in the north, we can see a sharp increase in body size towards polar waters, and in the opposite hemisphere, in the south, we don't have a clear pattern. So and also we can see in the contribution of different taxonomic categories, we can see that there is a large fraction of unidentified calanoid copepods because they are small and we don't have so much resolution in the image. So with our work, in this sense, we want to see clarify this pattern in the South Atlantic and basically resolve these identifying calanoid copepods in the image that have a large contribution in the WP2 size, which is the 200 micron size fraction in the plankton. So our goal with this work was to investigate body size distribution of most abundant copepod families and compare with environmental responses. So we want to test this classic overview in the latitudinal gradients, which is the Bergman rules, which want to see if there is an increasing contribution of large organisms in colder waters, high temperature, and also the additional influence of particular oceanographic features that we have in the South Atlantic, if it can increase, enhance this pattern. So in the South Atlantic, when we look to the oceanographic, we can see that we have two of the most energetic and high complex western boundary currents, especially here in the Southwest Atlantic. So we have the Brazilian current, which is a warm water that flows towards the south, which encounters the Malvinas currents, which is a cold water current. And it creates this region called subtropical confluence zone around 40 degrees. South of this region, we have subantarctic waters. And in the 6th degree, we have the Antarctic circumpolar current, which flows from west to east in a permanent direction. So we also, when we look to the South Atlantic, we have this large ecosystem that is called subtropical gyre that connects all the ocean, 3 oceans, so Atlantic with Pacific, and also Indian Ocean through the Agulhan currents that sometimes release some rings in this system. So this work, it integrates two expeditions. The first one is from Tara Ocean. So we selected samples from South Atlantic, mainly the tropical, temperate, and polar waters. And we have another cruise which was conducted in Brazil, in the south and southeastern of Brazil. So we can see a very nice large latitudinal gradient in the South Atlantic. And those samples were conducted in the end of 2010 and beginning of 2011. So we are also lucky that we have the same timing in both cruises. And so our sample, in total, we have 37 stations. The sampling was using a WP2 net, a 200 micron size. So the net was towed around 100 meters in a vertical tow up to the surface. So we also deployed a rosette with a CTD to collect the environmental variables, temperature, salinity, and oxygen, and also water to estimate chlorophyll concentration. So through my Assemble Plus action, I went to Villefranche-sur-Mer in France, and I have visited the zooplankton collection of Tara Oceans. So, and then we fractionated, sorted, and scanned at least 100 copepods. We sized them, and those high resolution images were imported to Ecotaxa to further taxonomic identification. And we have from this work, we have two approaches. I'm going to present the size-based approach, but we also identified to the species level the same individuals. So it makes possible integrate a microscope with scanned image. I could actually improve the taxonomic categories of identified calanoid because I was looking to the same organisms. So it was very nice. The size from the organism, they are in pixels, so we converted to equivalent spherical diameter in millimeters. So we measure the major and the minor axes of those organisms, and then we calculate the CD. So our data set, we could estimate abundance, body size, and biomass, and then we applied GLM models to see the relationship with environmental variables. So as a result, we can see through the four oceanographic process that we want to look at, that in BC, in the Brazilian current, we have a higher temperature and salinity. In the opposite, we have in the Antarctic waters, we have more oxygenated and more productivity waters. And in the confluence region and the subantarctic, we have more variability in the environmental variables. So we have more intermediated process. In the zooscan, this is the collection of families that we could find in the images. In total, we identified 25 families. And the ones that are in the red box are the ones that were more abundant. So we selected them to apply the models. In the latitudinal gradient of abundance, we can see that there is a large contribution in the Brazilian current of small copepods. So by this, we have families like Acartidae, Paracalanidae, Oitonidae, Corycheidae, Clausocalanidae, Oncheidae, that were families that are difficult to target from the image. And with our approach, we can actually have a better resolution of them. Then you can see that their contribution, they are dominant in these warm water conditions, and they can be reach 90% of the adult abundance. And in the copepodites, we can see that we have the most contribution of Clausocalanidii, which is a very important species in the South Atlantic. And as we go towards the cold water conditions, we have the dominance of Oitonidii copepods, and the copepodites, we can see that we have a large contribution of the Calanidii family. So the latitudinal gradient in biomass, we can see an increase in large copepods contribution. So especially Euchetidii, which are carnivorous copepods in the most tropical water conditions. As we go to polyward, we can see the increase of Hincalanides, the Hincalanides and Calanid copepods that are large copepods, and they dominate the biomass in those areas. But also, again, as Oitonides, they have a large contribution in the abundance. They also have some, they have in some samples a large contribution in the biomass as well. When we look to the community level, we want to test Bergman rules. We can see that abundance, biomass, and the body size, they actually increase towards subantarctic and Antarctic water, especially in those oceanographic features that we are looking at. So here in BC, in the transition of tropical and subtropical area, we have higher abundance. In the area of confluence and also in the Drake Passage, we have these Inyans in this pattern. And who is contributing to this community level pattern? Well, large calanoid copepods of the family Calanids, they buster this pattern. So they have a higher, as they have a large abundance, they have also a large impact in biomass in the subantarctic and Antarctic waters. Also, other families that are large in body size, they also contribute in increasing body size toward polar waters. And when we look to a small body size family, we have a very weak gradient, so they are small. They are small everywhere in tropical conditions, but also in polar waters. So in our models, body size, we have actually 3 patterns. So we can see that when we look to the family level, we don't have, we have 3 patterns. So body size can increase in some families in cold water conditions. There are some families that actually increase, that have the opposite pattern, they increase in warm water conditions. And we surprisingly also have families that don't have a linear trend, so they don't have a latitudinal gradient. It seems that climatic conditions that don't change their body size, especially the small families. Models of salinity was very similar to temperature, and with oxygen we can see the opposite pattern. So more oxygenated water have higher body size in some families. So our study gives support to global recent finds of temperature size rules in community level. However, not all families of copepods follows this rule. So this positive trend with higher latitude and low temperature is mainly driven by large body size copepods like Calanidii. And they actually they are driven by this increase in primary production in these areas in temperate waters, mainly driven by frontal polar waters, where we have more oxygenated water, more chlorophyll. So it's just a few families that follow this trend in the environmental variability. We have some families that have a very weak signal, so usually small copepods, and we think that this is related with some resilience to climatic variability. And in the end, we also see some families with inverse Bergman rules, especially cyclopoid copepods. So each family responds different to the thermal gradient, and this is mainly by the relative contribution of different life history strategy. So we see that these families of cyclopoid copepods, they have a large vertical distribution, and also there are some groups like Saferina that they explore different trophic groups, they are not driven by chlorophyll, or are not preferential herbivorous. So some species that are different, they are detritivorous or carnivorous copepods. So also I want to say that this body size approach was my first access to Assemble Plus call, but I have a second one that is going to be in the next month. So I'm coming back to Naples with this project that is called Assessing Species Boundaries, Connectivity of Copepod Populations in Transitional Climate Areas in the South Atlantic, we want to compare with the Mediterranean Sea. So we have samples from the Rebio from Arvoredo in Brazil, and we want to compare with the Gulf of Naples, targeting some copepod species to apply molecular tools. So those areas are climatic areas that are similar. So we want to target the same species in the same season to see how is the population connectivity. So I'm very happy to actually have a second access of using this funding from Assemble Plus. So thank you for listening. So this are the people who are my networking. So my supervisor here in Brazil, and also other university from the north of Brazil, Grazia from Stazione Zoologica, and also some people from the Villefranche, which was from my first access in the Assemble Plus. And I want to thank you all my funding support to my research. And thank you for listening.
Thank you, Erica. Very good. So any questions or comments from anyone in the audience? I have a simple question.
Okay.
You may have, it probably was in the slide, but I didn't quite. So what is, let's say, the range of sizes related to, the Bergman effect? So you're talking about 50% bigger if it's high oxygen, or what kind of differences are you looking at?
You mean the the contribution of each size fraction?
No, you were talking about the oxygenated waters. The copepods were larger.
Yes, yes.
Yes. So my question is, how much larger? What kind of proportions are we talking about here?
Okay. So we can see by size, usually in warm water conditions or in the Brazilian current, we have species, average size of the species are 1 millimeter. So when we look to calanid families, they have 3 millimeters of size. So they are 3 times. Sometimes when we have the Hincalanidi contributing to biomass, they have 4 or 5 millimeters. So sometimes it's 5 times larger than the average in the tropical waters. So
okay, so yeah. But I guess there in those waters there's plenty of oxygen. Yeah.
Yes, yes.
So essentially,
because
this is what
it's
happens.
a highly we have more variability in temperature, oxygen, everything, because of those areas down to 40 degrees. We have various sets of frontal system. So this actually enhance the oxygen contribution to this area. So it's not only the temperature that matters when we are looking to size. Also, there is a large gradient related with oxygen in this.
Yeah, probably oxygen is the key factor, because those waters tend to have more oxygen. Very good.
Yeah. So thank you.
Yeah. Okay. Thank you very much. I don't know if there's any other question. If not, thank you very much, Erica. Very interesting work. And well, we hope to see you again, maybe in a next assemble or something like that. Anyway, keep your eyes posted on the internet, because there'll be news. I think later in the year we'll have more programs for access, which will be already independent from assemble, but there will be opportunities for people to continue on this track.